Hello campers, this is Dr. Cary Harding from Oregon Health and Science University. I'm sorry I can't be with you at camp this year. Hopefully next year I'll be there, but it's my honor to give you a bit of an update by Zoom. So today I want to give you an update on liver gene therapy, both a little bit about what's happening in the laboratory, but as well on some uh, clinical trials. Our laboratory research most recently has been funded greatly by funds from the National PKU Alliance, but we do receive base funding from the National Institutes of Health. I do have a couple of financial disclosures, and with regards to this presentation, the things that are most relevant are the two clinical trials that I'll talk about, which are funded by BioMarin and Ultragenyx. So these are my objectives, although some of this will be review for people on the call. I want to review the cause of PKU and how PKU damages the brain. We'll talk about gene therapy experiments in PKU mice and early clinical trials now in humans. And then we'll talk about a novel gene insertion approach, which has not been tried in humans yet, but that we hope will work in anyone with PKU, regardless of their PAH mutation. So this is my favorite enzyme, phenylalanine hydroxylase. Normally that enzyme takes phenylalanine and converts it to tyrosine, but when it's not functioning normally, the phenylalanine builds up in the body and it can be converted into phenylketones. The phenylketones show up in the urine, and that leads to the name of the disease, which is phenylketonuria. And left untreated, this inherited disease, besides causing high blood phenylalanine, can lead to these other clinical problems, the most severe of which being developmental disability. We don't see this very much anymore because of the effectiveness of dietary therapy. Now the enzyme resides mostly in liver. Excuse me. But when the enzyme's missing, the liver doesn't particularly care. It doesn't cause the liver any problems, but the elevated phenylalanine in the blood then crosses over into the brain and causes brain damage. We understand quite a bit about how phenylalanine causes brain problems, but we don't completely understand all of it. And I actually have a separate NIH grant to continue to evaluate the mechanisms behind how phenylalanine damages brain. Suffice it to say, we do think that or we are pretty certain that it is related to elevated phenylalanine, and anything that we can do to lower the blood in the brain Phe will prevent the major complications of the disease. So there are actually several new treatments that are in various stages of investigation that lower blood and brain phenylalanine and don't require dietary or as much dietary phenylalanine restriction. But I want to focus on what I call the liver-directed genetic therapies. And just to review, I mean, the cause of PKU is that the individual has changes or variations in their PAH gene, which is the code for how to make PAH protein. And these two changes in the genes cause variation in the message produced by the gene or the messenger RNA. The messenger RNA then leads to the production of it either causes the production of a variant protein that has no function, or in some cases it just completely lacks the production of any protein, and then you cannot convert phenylalanine into tyrosine. So how do we deal with this situation? Well, sort of working in reverse, one potential possibility is that we could supply the message, could put the messenger RNA back in. But RNA is very unstable. It has to be stored at extremely low temperatures. If you just inject it, it gets rapidly destroyed by enzymes in the blood that are called RNases. So we have to protect it. And so we wrap it in a lipid nanoparticle, gets injected into the bloodstream, and the lipid nanoparticle carries it to the liver. And in the liver cells, the messenger RNA gets translated into PAH, where it can then catalyze the reaction of phenylalanine to tyrosine. This is precisely the method that's used in the Pfizer and Moderna COVID vaccines, except in that situation, the mRNA is the code for the COVID-19 spike protein, and the lipid nanoparticle targets muscle cells. In our case, so then the COVID vaccine carries the messenger RNA into muscle cells. The muscle cells make a little bit of the COVID spike protein, and that's enough to stimulate our immune systems to recognize the COVID virus and destroy it. In our case, we want PAH messenger RNA to be delivered by a different lipid nanoparticle that targets the liver, not the muscle, and to keep delivering the messenger RNA to make PAH protein. The drawback of this approach is that probably has to be delivered at least weekly, maybe twice a week, as the message is not going to be permanent and has to be repeatedly administered. But Moderna actually is actively investigating this, and they just presented some data at the American Society of Gene Therapy meetings a week ago, showing quite nice correction in PKU mice, giving them weekly injections of just such a product. Switching to the next possible treatment, gene addition. So this is sort of backing up one further step. If the RNA is unstable, we can deliver DNA, which is actually quite stable and very easy to handle. The DNA is the code for how to make the messenger RNA. And there's a variety of ways of delivering this to liver, but in our the most common way to do this is to take advantage of nature's best gene delivery vehicles, which are viruses. And one of the most popular viruses to be used is a relatively harmless virus called the Adeno-Associated Virus, or AAV. We package our PAH gene in AAV, deliver it to mice and or in humans, hopefully, and it will carry the gene into the liver, and then the gene will make messenger RNA, and the messenger RNA will make PAH protein. But this approach doesn't actually do anything to correct the patient's own mutations. The gene doesn't interact with the genetic material in the patient's liver. So how could we permanently correct the patient's own genes? That's a method called gene editing. I'm not going to talk anymore about the lipid nanoparticle methods, but we will talk a little bit more about gene addition and gene editing. So a bit more about gene addition. We have been working on this for several years in the laboratory, and I've talked about it at camp before. There's various flavors of adeno-associated virus, and the one that we've used most commonly is something called AAV8. And this takes advantage of or models a system that has been used in adult males with hemophilia, which is a clotting disease. This is just a report that's several years old now, that after a single infusion of AAV8 in these individuals, they start making clotting factor in their blood, and some individuals are almost 14, 15 years out now from a single infusion and have been pretty much cured by this single viral infection. So how does this work? We have an AAV8 that we have engineered to carry the PAH gene. This is a little picture of them on an electron micrograph. It's the little tiny particles in here. They go in and infect the liver and carry the PAH gene in with them, and they start making PAH protein. And again, they don't interact with the PAH genes that are in the mouse or in the human. We reported on this all the way back in 2005, and we're not the only lab that has done this. There's been other labs in the world that have successfully accomplished the same thing. And when we inject this in PKU mice, their blood phenylalanine levels plummet down into the normal range. And one nice thing about this is PKU mice are hypopigmented. They're tan instead of black, which they genetically should be black. And they're not albino, but the high blood Phe lowers their pigmentation a little bit. And when we lower their blood Phes, their black color reemerges. So we can tell pretty much in the cage who is cured just by looking at their coat color. So why hasn't this been translated into the clinic sooner? Well, we've obviously had a pretty decent treatment and the disease isn't life-threatening. So for safety reasons, we obviously wanted, we wanted to prove that this treatment was safe before we ever did to take this into humans. And I had the pleasure of being involved with another trial, a disease that is life-threatening and where the treatment was not nearly as effective as the therapy for PKU. This is a disease called ornithine transcarbamylase deficiency, or OTC deficiency. And we have treated 11 adults and have just presented this data at the ASGCT meeting again a week ago. And in these individuals, the best news is that the treatment has been proven to be extremely safe. They've had a little bit of liver inflammation, which we've learned how to treat with a brief duration of oral steroid treatment. And really good news is that 7 out of the 11 patients have responded to the treatment and have either reduced their dependence on the low protein diet or have completely eliminated their diet therapy. And in some cases have been able to completely come off of the medications that they have to take for this disease. And one of these responders, has been able to completely come off treatment and has been off treatment for 4.5 years after a single IV infusion of this virus. And the company, Ultragenyx, which sponsors this trial, is starting a phase 3 trial at the highest dose of virus that we used in 5 of the 11 patients. So this has been a very satisfactory result, and hope that we can continue to achieve good results as we go forward. So based on these types of safe and promising outcomes, there are now 2 ongoing liver-directed trials in adults with PAH deficiency, one run by Homology Medicines, the other one by BioMarin Pharmaceuticals. They both have their own flavors of AAV, one called AAV-HSC, the other one AAV5, and I don't have any prejudice or information about which one's necessarily better. There isn't any really any much data out there yet on their effectiveness. There's not been any significant safety problems from either infusion. Homology's had a couple of press releases showing that 2 of the patients have responded with blood levels that have dropped below 360. And BioMarin hasn't released any information yet. So more information to come on the results of these 2 first gene therapy trials in adults with PKU. So there are some issues with this approach. As I mentioned, the genes that we deliver don't interact with the genomes in the liver. And if those cells divide for some reason, the viral genomes get lost. And we know that this does not work in the juvenile liver. And so in children or in infants, the livers are growing so quickly that the viral vectors get kicked out. So it won't work in children. And we don't know if this is truly durable in adults. It may be that over years and years it's slowly lost because the liver does gradually turn over. At very, very high doses of AAV, there are more significant inflammation and immune responses to the AAV virus, which can be problematic. And although I've told you so far that the viral vectors don't integrate, that this is actually not entirely true. There's a very small number of cells where the viral vectors the viral genomes do actually insert themselves into the liver genomes. And so there's a theoretical risk of liver tumors developing, although it's not been seen in clinical trial yet. So there's a ongoing discussion about whether or not there's a risk for this. So what about being able to actually correct the patient's gene back to the normal sequence? There's a variety of ways of doing this, but one of the most widely investigated ways right now is to do gene editing with a system that's called CRISPR-Cas9. This system was awarded the Nobel Prize back in 2020 for 2 of its developers, Jennifer Doudna and Emmanuelle Charpentier. If you want to read more about this, Walter Isaacson has just recently published a biography of Jennifer Doudna. I haven't read it yet, but Walter's a quite good writer, and I'm sure it'll be a very entertaining read. So how does this work? Well, what we've done in the PKU mice is we've taken our same AAV8, but instead of the PAH gene, we've put in the gene for Cas9. So what's Cas9? Cas9 is a protein from bacteria, and it's part of their defense mechanism. So bacteria get infections by viruses too, just like we do. And Cas9 developed over time, evolutionary time, excuse me, to recognize foreign viral DNA and destroy it before it hurts the bacteria. But what Dr. Doudna and Dr. Charpentier developed was they were able to tweak this system to be able to teach Cas9 to recognize very specific DNA sequences in mammalian DNA and human DNA, and teach Cas9 to cut at very specific sequences in our genomes. And that's the beauty of this system. It will cut at only the place that you teach it to. So I'm leaving out some of the details of the system here, but essentially what we've done here is to teach Cas9 to go into the PKU mouse liver and attach to the site of the mutation in the PAH gene and make a cut. Now, I think the lay press and even people in the field have somehow gotten the false impression that Cas9 can cure genetic disease. It doesn't actually cure anything. Cas9 actually makes genetic disease. It just goes in and makes a cut. Well, that's just deleting the gene, which is essentially, making PKU. And in fact, Dalen Richards, in my lab has used this system to make another different type of PKU mouse. So Cas9 doesn't cure anything. And I'm just showing it here, cutting one of the genes. It will cut both of them, but I'm just for simplicity showing it cutting one. So again, it doesn't cure any disease. It makes a cut. So how do we make it cure the mutation? So what we have to do for that is we have to actually use a second virus and deliver a piece of DNA that matches the gene but has the normal sequence. And what we hope is that the cell will actually use that repair template, we call it, to fix that hole. So the cells don't like having holes in the DNA, and it will do anything to fix it. And we hope that it incorporates this normal piece of DNA, incorporates that, and then now you have a normal copy of the PAH gene, and now we'll make normal PAH enzyme. What Dalen Richards learned, though, when she first did this experiment is the cells actually prefer to just take the two ends of that hole and just stick them back together again and ignore our repair template. So it's fairly frequent that the liver cells just don't use our repair template. So we just end up with what we call a deletion and don't have our repair in there. So she had to use a trick to get this to work. And I don't have time to really explain it in detail how this happened, but she was ultimately able to successfully to get about 13 to 15% of the liver cells to incorporate our repair template. And when she did that, those cells gave us about 10% of the normal amount of enzyme activity. And in those mice, they had lower blood phenylalanine than animals who didn't have that amount of gene correction. Now, the phenylalanine levels had come down a lot, but they didn't come down to, on average, down to the treatment level of 360, which is in red, or down to normal, which is 120 in black. Although a couple of animals got close to that, but on average, they're around about 600 or 700 micromolar. So promising result, but not quite the cure that we're hoping for. So what we learned is that the frequency of these corrections is just still a little bit too low, and we need more of them. And in order to really have this be successful, we need to increase the frequency of the initial correction. Or the second possibility, which I'll talk about in a minute, is to give the cells that have gotten corrections a bit of a selective advantage to get them to grow and repopulate the liver and expand into a population that becomes a bit more relevant and can uh, become a large enough population that they'll lower the blood Phe even a little bit lower. Now, the other issue that we have with this is that the reagents that we designed were specific to attack the mutation in the PKU mouse. There are over 1,000 different mutations in humans with PKU. So theoretically, we would have to design reagents for every different mutation, and that's just not very feasible. So could we develop a set of reagents that would work for every individual with PKU? And that's a strategy that we call gene insertion. So we make the same kind of double-stranded break with CRISPR-Cas9, but instead of just using a repair template that strictly matches the gene that we're trying to repair, now we're trying to insert an entire copy of the normal PAH gene into that hole. So regardless of where the mutation is in the patient's gene, we're going to essentially replace their whole gene with an entirely functional copy of PAH. So it doesn't really matter where the patient's mutation is at. So that's the goal of the current approach that we're working on. And we've had a little bit of early success with this. But because we're trying to put in a piece of DNA that doesn't entirely match the region that we're targeting, this is not quite as favorable as what we were trying to do before. So we've just reported on this at the ASGCT meeting a week ago. We've had a little bit of success with this. We've gotten enzyme activity of about 7-8%. And in those animals, their blood Phes are significantly lower than in untreated mice. But again, the mean is, again, not down to the normal range or the treatment range, but it's dropped down to about 1100. But we still have a lot of work to be able to raise this, the frequency of this happening up to a level where this is relevant. So there are some CRISPR trials going on in humans. The first two instances of this in blood diseases and in lung cancer are taking bone marrow cells or white blood cells from the blood and actually doing the editing out in the Petri dish and then reinfusing them into the patients. The first instance of actually injecting CRISPR reagents directly into the body is being done here at OHSU at the KCI Institute, where they're putting CRISPR reagents into the eye, but they're not using a repair template. They're just trying to knock out a gene that's actually a toxic gene. So they're just, again, using CRISPR to cut the gene and knock it out. And then the first instance of actually doing CRISPR in liver is the same sort of thing. They're just having to knock out a gene that has a toxic effect. And they're not using a virus in this situation. They're actually using a lipid nanoparticle to deliver Cas9, just messenger RNA. And the Cas9 goes in and makes a cut in that gene and knocks it out permanently. And that's also working quite well. But no one's actually attempted a repair in humans, as best I know, just yet. So as I mentioned, one possibility is if we don't have very many cells that have been edited, can we actually select for them and make them grow to a population that's a more substantial, relevant population? And we've had a collaboration with Dr. Marcus Grompe's lab on our campus, as well as with this particular project is with a lab in Italy run by Dr. Luigi Naldini. And this work was led by a graduate student in Marcus's lab, Ann Beneda. And what she was able to show was she was able to give cells that have gotten a gene insertion a selective advantage by making them resistant to toxicity from Tylenol. So normally, if you take too much Tylenol, your liver cells die. But she made the cells resistant to Tylenol if they have successfully gotten a gene insertion. So in the line here in blue, these are animals that have gotten a gene insertion. She's given them Tylenol. And as the other cells in their liver have actually died from Tylenol toxicity, the cells with the gene insertion have expanded and they're making PAH enzyme activity. And over time, their phenylalanine levels have decreased. And so within about 3/4 of a year, their whole liver has been taken over by PAH expressing cells. So this selection is actually working to expand the number of edited cells in the livers of these mice. So between methods that we're using to try to increase the initial frequency of gene editing and potential selection methods, that like this acetaminophen selection method, we hope that in the future we'll develop a robust way of actually permanently curing PKU. This actually obviously takes an army of people, and I don't have everyone listed on this slide to do this work. Dalen Richards, who's graduated from my lab and has now gone on to medical school, did the initial gene editing work. Michael Martinez is a current graduate student in my lab who's continuing on with this, and there's several other laboratories that we have collaborated with over the years. Again, I want to acknowledge the funding that we received from the National PKU Alliance and the National Institutes of Health, and I want to thank you for listening today.